Okay guys, here we are back with the second part of PCR cloning. If you remember where we left off last time, we had designed primers to amplify a specific part of the PLC gamma SH2 domain. In this case, just we amplified, let me scroll down here so you can see it, this segment of the um, PLC gamma. So from here to here, we actually got the wrong places here to here. We amplified that DNA using the primers, which are shown with the symbols um, underneath here. There's the left primer, and then here on the bottom is the right primer. Okay, now what do we need to do is take these primers and use them to clone this piece into the proper plasmid. To do that, let's come back up to the top here and look at the primers. The primers are fundamentally different from each other because they bind to different strands of the DNA. The left primer, we don't have to do anything with it. There it is, CCAATT. And like we talked about, this you can find in the sequence. Okay, so what I'm going to do now is build this to get whole DNA piece together using Microsoft Word. So I'm going to copy this sequence and go into Microsoft Word. So here is the left primer. And it starts with the 5' end. There's the primer, and then the 3' end. The right primer, whoops. So actually I'm going to copy this entire document and put it in Courier format. That's the font that we need, Courier, because it makes all of the characters the same size. Okay. And then the right primer. I have to go back here to grab that right primer. And it also is from 5' to 3' here. But you'll see in a minute that the right primer is going to cause us a little bit of trouble. But we can figure it out. Now we are going to clone into the BSRG1 restriction site. And so BSRG1 has this restriction site, TGT-ACA. And then it cuts between the T and the G. Okay, so I'm just going to put this up here and indicate that like that. So we have to build that restriction site into our primers. So let's take the left primer and look at it. It goes CCAATTGGACA. So actually we're going to be lucky with that one. Watch what I do here. So you take the primer CCAATTG, and I'm going to add a T here instead of that G. Then it's ACA, so I don't have to change anything else. So we're super lucky. That hardly ever happens. Now I'm going to highlight that T right there in green, and then come back up here and that G. So the G has been replaced with a T. Now you would do this in a real live lab, and you would just order the primer with the restriction site built into the primer, not necessarily the sequence that's in the DNA. We're going to do the same thing with the other one, although we're not going to be nearly as lucky. Also another point, since this one's the left primer was so easy, I made it in the middle of the primer. In most cases you want to be closer to the 5' end, and so notice we with the right instead of the 3' end, because the 3' end is where the DNA will get made. So the CACGGGCTCC, we don't have a whole lot of leeway there. I'm going to make some changes here. We'll try and conserve one of those G's and one in the C. Okay, I think I can do that. Let's type the sequence out and then make the replacements Oops. So that this is the confusing part, and or one of them. Okay, there's the sequence. That's what we got out of the um, Primer 3 software on Biology Workbench. And so we need to change it to TGT-ACA. Now if I change that T to an A, that's an A, that'll be a T, that G will be left alone, then we have a T. So the changes here, I'll make them in yellow, that we had to make were that C to a T, okay, that A, there. So we had to make 4 nucleotide changes, and I'll reflect them up here in the original sequence. That had to be changed to a T. That G was changed to a T. That was changed to an A. Oops, I didn't mean to do that. That G and that T. Now you might be a little bit confused, but just forge on ahead. All we've done now is take the original sequence and then make changes to make primers. Okay, let me save this real quick. Now, the next thing that we want to do is amplify this sequence from the PLC gamma sequence. So I'm going to go grab that and paste it in here too. Here we go, we're back. So let's find this left-handed primer first. We don't want the one with the BSRG1 site. We want the original sequence right here. I'm going to use the Control F function, and there it is. It finds the primer sequence in my DNA, and let's highlight that a certain color. Now we need to go up to this one. This one is a little bit different. If you search for this one, you're not going to find it in the sequence, and that's because it is on the complementary stranded DNA, not on this single-stranded piece shown down here. So we have to take what's called the reverse complement of the right primer. And let me do that right now. So this is called the reverse complement. Now you're going to have to study this because this confuses people, but you can do it. Now the 5' end of that is starting, I'm going to start here. See where I was at the 3' end, that G. On the opposite strand, that would be binding to a C, and then a T, and then an A, and then a C, and then a T, G, A, G. Whoop. Now, you may have followed this or you may have not, but I started at this G and I started doing the complement. So G binds to C. The next one is an A. A will bind to T. Next one is a T. That will bind to A, etc. Now we should be able to take this reverse complement and search for that and find it in our sequence. And there we go. Thank goodness. There it is. So now we have found the 2 regions in the DNA that the primers bind. And so I'm going to grab those for clarity. Oops, I don't want to get that part. And then I want to copy them out. Down here, just so we can follow along with what's going on. And I'm going to call this the PCR fragment from the PLC gamma. There it is. Now, this sequence here has not been altered, but in real life, if we did this, let me zoom back up, we are using these guys. The ones where we changed it to have the restriction site. And so we need to build in TGT ACA down here. With this one, it was pretty easy. It was CCATT, and then instead of TGG here, it was TGT. So now we're done actually, ACAT. So that one was easy. This guy here on the bottom is going to be a little bit harder because it's the reverse complement. Whoops. Let's go back up and look at this sequence. The reverse complement here, well, the right one is CATGTA, but we have to flip it and go the other way. Now, I know it's these. Let me take this off so you guys can see. Here's the restriction site right there. See that TGT-ACA? Now we have to find it up here on the reverse complement. And so instead that's these last two here. The GT binds to the CA. And then here, instead of this little stretch of 6 nucleotides there, 1, 2, 3, we have to change that to read. It's going to be the reverse. So that'll be TGT-ACA as well. And so we need to change that in the sequence down below. So I'm just trying to get a handle on where we change it. So that G, that's where it starts. So 1, 2, 3. Okay. So let me go down. Right there is where it's going to go. And I'm going to copy back TGT-ACA. So the restriction site goes right there. So now we've done the changes. The next step is we need to clone this into the PRSet PAGFP plasmid. And in order to do that, we have to cut the PCR fragment with the restriction enzyme. So cut PCR fragment with the BSRG1 enzyme. When you do that, I'm going to copy this. You get exactly the same thing except for where it cuts. And if you remember, I told you it cuts between the T and the G. So now look up top here. If you cut, it would cut between that T and G. That's where the enzyme cuts. So in reality, what would happen is we would lose those. Down here, we find the TGT-ACA. It's right there. And so if we cut, we would end up losing those. So we now have the shortened PCR fragment that we can clone into the vector. So I'm going to go grab the vector. I'll be right back and we'll do the cloning and see what happens. Okay, I'm back. Here is the cloning vector, PRSet PAGFP. So what do we need to do here? What do we have to do? Well, remember we want to clone this PCR fragment into the BSRG1 site. The BSRG1 site is TGT-ACA. So we have to find that in the PRSet plasmid. So I'm going to search for that. And there it is, TGT-ACA. Let's make that red so we can find it. So that's our restriction site. Now, just like we did last week with restriction enzymes, we're going to cut this, and it cuts between the T and the G, so we open it up. Whoops, open it up there. Then I come back up here, and I grab my PCR fragment, copy it, and paste it into that spot. So there we go. Now, one thing that's sort of cool is notice here the T that's red at the very beginning here of the insert comes from the vector, and then there's the restriction site has been restored, so it goes TGT-ACA. Let me highlight that and change its color so that you can see it. Now the restriction site is back. The same thing down here. In blue there's a T, and then the GT-ACA is in red. But now we have the restriction site again. So when you clone into it, you restore the restriction site and you gain another one on either side of the insert. Now we need to analyze this. And So I'm going to copy it, the whole new structure, and go to BLAST. So here we are at BLAST on NCBI. We want BLAST X because we're going to put a nucleotide sequence in and search the protein database. There it is. So this is the PR set PAGFP with the inserted PLC gamma into the BSRG1 site. Okay, then I'm going to search the non-redundant database by BLAST. Okay, there we go. We've gotten the results back, at least in the conserved domain database, showing us the picture. Notice there's GFP, and then in frame is the SH2 domain from the PLC gamma. So that's pretty cool. We got it on the first try here, so we don't have to change the frame. If you follow down here, there's the beta-lactamase. That's since we copied the whole plasmid. That's the protein that makes the antibiotic resistance. And so that's how you clone by PCR-based cloning. It's a combination of doing amplifying a piece of DNA by PCR and then cloning that into a vector using the restriction enzymes. So it uses some of what we learned last week as well. The hard thing here is designing the primers with the restriction site that you want and then changing those primers. The left-hand primer is not so bad, but the one on the right can be confusing. So you have to go through very slowly and step by step. First, take the primer, then take the reverse complement, and then add in the restriction site. Okay, so now everyone, let's go look here at the homework that you're going to do for this particular exercise. So here's your homework, cloning by PCR. I want you to subclone any domain from your favorite protein. And you can use the one that you just used for cloning by restriction digestion. And I want you to do it by into the PGEX vector system, not into this PAGFP one. The PAGFP PR set one is much more difficult because it doesn't have the 3 frames. So we want to tackle that problem later. Right now, I want you to use PCR to amplify your fragment. You can build in any restriction enzyme of your choice and clone it into the PGEX system. Okay? Clearly show me the steps that you take, showing me the neb cutter results, etc. Do it in a stepwise logical fashion and show me how you're doing it, and then clone it into the PGEX vector 1, 2, or 3. And then show me the blast results and the sequence of your resulting fusion protein. Okay. Now conclude the homework with a description, and I just want you to briefly give me a paragraph or so telling me what you feel are the advantages and disadvantages of restriction digestion cloning versus PCR-based cloning. And that's your homework. It's due next Friday, June 27th. Okay, thanks guys. Bye.